Hello, everybody. In today's video, we will discuss the basics of microbial growth. So let's get right into it. By the end of this module, you should be able to discuss the basic differences and features of microbial growth in a laboratory versus in nature, define binary fission and compare it to budding and spore formation, calculate generation time for a bacterium, and outline the features of the 4 stages of bacterial growth in a closed batch system. All right, so with that, let's get started. So to start out, in nature, microbes show this kind of dynamic and complex form of growth, and oftentimes that's in biofilms. And we've talked a little bit about biofilms coming up to this lecture. And so however, we oftentimes are actually growing microbes in the lab, and what we learn about how microbes grow is based on growing them in the lab in a system that is completely different from the way that they grow in nature. We grow them in pure culture in particular. Um, now, when a microbe's nutritional requirements are met and they are able to, and they are in an environment with within like growth parameters that are within their ability to grow, then the microbes like actually grow in size and then divide. But that's not what we think of as growth when we think of microbial growth. We're actually talking about cell division. And so the process of one microbial cell dividing into two new daughter cells is what we consider to be microbial growth. And we've actually calculated that in class to some extent, or at least we've calculated microbial populations doing the different plate count techniques that we've done. And but when this all comes to like discussing how microbes grow in nature, it gets a little bit hazy because our numbers are based off of this very few number of microbes that are actually able to grow in the lab, which is actually somewhere around 1% of bacterial species. Now, that means that there's 99 to 99 point something percent of the bacteria out there that we have never grown in the lab, and but they do exist in nature. And so we're going to try to think about how that might be a little bit different here in just a moment. But keep in mind that for much of this, we're not actually able to see it. So this is an example of a streak plate that someone has done. And you can see that we ultimately get these individual colonies. Now, this plate appears to have two different types of organisms on it. We've got these kind of white colonies that are nice and circular or yellow. And then we've got these really light white colors that are a little bit less circular, a little bit irregular, a little flatter, a little duller. And so those are probably different organisms, but they're both growing on this blood agar plate. And so while there is a little bit of intermingling going on here, um, ultimately, if we were studying these in the lab, we would separate them onto their own individual plates. And if we wanted to know about both of them, then we would grow them on separate plates and do the assays independently. But in nature, these bacteria are going to be intermingling not only between these two, but oftentimes between multiple other organisms, including archaea and fungi and protozoans and all sorts of stuff that are out there in their environment. And so there are going to be many different environmental factors that actually play a role in how those microbes grow and whether or not they're able to perform one type of metabolism or another, and even in the phenotypic characteristics of those bacteria. So, for example, E. coli is generally a motile bacillus shape. So that means that they have these paratrichous flagella, they swim around in their environment, but they convert to these filamentous non-motile bacteria when they're actually causing UTIs. So E. coli are the most common cause of UTIs. They're fecal bacteria. And so if E. coli gets into the urinary tract, it's very likely to cause an infection. And that what's interesting about it is that they don't look like these cute little swimming bacteria anymore. They grow into these really long filamentous rods when they're in that urinary tract. All right. Um, Now, speaking of UTIs and other diseases that can have some sort of indwelling device, many of these are actually subject to the formation of biofilms. These biofilms in medical settings may oftentimes be caused by a single organism. So this can be either a catheter, which could lead to a UTI, or maybe a replacement heart valve or a replacement hip um, joint or something like that. When you get these, it's very possible to actually get a biofilm formation. And those biofilms are difficult to treat when you have bacterial growth and biofilm formation for multiple reasons. One of which is the fact that the biofilm itself, that polysaccharide, can actually prevent the transmission of antibiotics. Oftentimes, these areas may not actually be easily accessible by the immune system anyway, and so it can leave, it can be difficult to treat. Now, this is similar to what we might see in the environment. Oftentimes, in environmental settings, the microbes are actually growing in biofilms, and these biofilms are sometimes single organisms, but they're oftentimes actually a multi-organism or a multi-species community. And it may even span kingdoms or domains. So you might have archaea and eukaryotes with bacteria in the same biofilm. When these biofilms are formed, they can organisms can communicate between each other to help collaborate and survive. Um, they also, both for the communication during the biofilm formation and after formation, it may actually be triggered by the gene regulation that can come with quorum sensing. And so keep that in mind as a link back to our gene regulation lectures. Now, when microbes are actually dividing, this is the process that we think of as growth. It is usually going to be done by binary fission. And this is a single cell, which we see a bunch of individual cells in this false colored SEM image. You will see a single cell that will actually divide into two daughter cells. So we see the formation of two daughter cells here. Now, this process is asexual, so we're just duplicating what's in the single cell and making it into two cells. This is the process that we've talked about for DNA replication and all that stuff in the past. Now, when the parent cell begins to pinch off, which we see from this arrow pointing at this septum that's being formed, you're going to actually see this start to form, and then it pinches off. This is through the formation of these proteins called FITZ, FTSZ, or FITZ ring, which are involved in this cell division process. And these two cells at the end will become genetically identical. Now, this is in contrast to budding. Now, there are some bacteria that bud, and this is very common in yeast cells. This is another form of asexual reproduction. In this case, you have a single cell, which are these big guys, that will start to develop these small outgrowths. So you can see that there are multiple buds coming off of many of these cells. And then the chromosome is going to be duplicated and put into that bud. And once the chromosome is put into that bud, the bud can then separate from the mother cell and then, grow into a full-size cell. This happens in some bacteria. It definitely happens in several fungi. And so it's a little bit different than binary fission. It's not like a complete elongation and then like an equal sharing of size and components. But it is still a form of asexual reproduction. Now, you may also see some spore formation. Um, in my experience, spore formation is primarily used um, for going into dormancy, and so you're not actually getting like a duplication per se. Although I guess there are some species that may just form spores that they actually release into the environment that can later germinate. And so spore formation is common amongst fungi and some bacteria. And fungi, we've talked about this. There are sexual and asexual types of spores, but in bacteria, it's always asexual. There is no sexual reproduction in bacteria that I'm aware of. Now, what we've talked about with spores in the past are these the dormancy style spores, where you get this um, thick wall of cell wall that is kind of blocking it off and protecting it from the environment, and it's not growing. And this is something that you see like in these cells over here. We can see the formation of these spores inside the cell. And usually what that means is that the actual rest of the cell is just going to shrivel down to nothing, and all the important stuff's in this little circular area here. But I guess in some species, like these Streptomyces species, they actually form spores that hang off the ends of their these like hyphal-like extensions, and those can be released into the environment and later germinate. And so that brings us to thinking about microbial generation time. So generation time is a pretty important concept. This is how long it takes for an individual cell to completely divide and form two cells. And the amount of time it takes for this to happen can be largely variable. And for example, under ideal growth conditions, ideal temperature, pH, oxygen, nutrients, etc., E. coli can divide in approximately 15 minutes, 15 to 20 minutes. Some species of Mycobacterium, that is the species or the organisms that cause stuff like leprosy and tuberculosis, they may take 24 to 48 hours to have a full cell division. And of course, the amount of time that it takes for the cell division to occur is going to be determined not only just by the individual species, but by the environmental conditions that they're living in. E. coli is going to divide much more fast in a 37 degrees C environment than it is in a 10 degrees C environment. Now, in this example down here, we're showing you how to calculate generation time. This is growth time in minutes divided by the number of generations. So if we start with a single cell here, then we have 1 generation, 2 generations, 3 generations in the period of 1 hour. Then the way we calculate this is the number of minutes, 60 minutes, divided by the number of generations, 3. So that gives us an average generation time of 20 minutes. When you do something like this, it's always a good idea to have a couple of iterations of generations to get an accurate number, because there can be some variability. But in terms of like the kind of stuff that I'm going to ask you in class, we would just assume that each of these is a 20-minute interval. And so, as I was saying, nutrient availability is going to impact how fast microbial populations increase. We have something like E. coli here, which, as I mentioned, about 20 minutes to reproduce. And then you have the slower things like Mycobacterium, which here it says 15 to 20 hours. Under non-ideal conditions, it could be even longer. Now, we usually grow organisms in batch cultures, right? So either in a broth or on a plate. And in broth especially, we can see this very clear like growth curve that occurs. And they have these distinct phases. So these 4 distinct phases. And these phases are going to be important for you to understand, because well, put it simply, we're going to ask questions about each of these different phases and what's going on there. All right. So Let's skip that. We're going to go here. We've got phase one, which is the lag phase. So on this graph, this is right here. So if we take an individual microbial cell and add it to a broth, it doesn't immediately start dividing if it was not actively dividing before it was added. So if we take, say, for example, a culture out of the fridge, our cells are not probably growing at this point. They are just hanging out. So we put them into a broth culture and add them into a nice, warm incubator. We don't immediately start seeing division. It starts out with this, what's called the lag phase. And this is the delay in growth where we're not seeing binary fission taking place. And what's going on is basically they're turning everything on. They're adapting to this new environment. They're like, okay, we have a bunch of nutrients available. We need to start turning stuff on and duplicating because it's time for us to replicate. And that's followed by the second phase, which is our log phase. The log phase is called the log phase because you get exponential growth. And so one cell after one generation becomes two, and after two generations, 4, 8, 16, 32, 64, 128, 256, 512. And after 30 generations, which if you're doing a generation every 20 minutes, that's something like 10 hours, you can have millions of bacteria coming from one individual cell. Now we come into the stationary phase. At this phase, usually if we actually plotted this, we would see that the line isn't exactly flat. It's more kind of like ebbing and flowing up and down, kind of like the red dot is here. So cells are still actively growing, but nutrient levels are reducing and waste products are accumulating. And so that means that your growth and death rates are approximately equal. Eventually you get to phase 4. And in phase 4, this is the death phase. This is when you reach this critical buildup of waste, right? And when you get to this critical buildup of waste, you don't have very many nutrients left. The cells just start dying and you get exponential death or exponential decrease. You will still have some cells that survive at the end of all this. These are cells that either are able to have some sort of adaptation to feed off of the waste products that were produced, or they're able to, have some sort of mutation to deal with the influx of waste products. So they're not usually all dead at the end, but they may be like dormant or slowed down in growth. And so that's why in like industrial settings, we're going to kind of regulate this. And so what they do is they often use something like a chemostat. In this case, you have your air inputs that go in and out. So you can pipe air in here to oxygenate, and then the excess air will come through here. And then you can add in fresh media through this. And this is when the fresh media, or when the media comes up above this line, it will exit through here. You stir it around to keep everything mixing. And so what this does is it allows you to have a continuous culture of whatever you're trying to grow. And this is what they do when they try to produce microbially produced products. You're constantly feeding in new media and removing excess old media, which is going to be a mix of the waste products and the dead cells and some living cells, and all that gets flowed out. And then from that, you can also, extract whatever you're trying to get from your chemostat. All right. So at this point, you should be able to, I'm going to go back to the beginning because I didn't copy it to the end, discuss the basic differences and features of microbial growth in laboratory versus in nature, define binary fission and compare it to budding and spore formation, calculate generation time for a bacterium, and outline the features of the 4 stages of bacterial growth. I hope you enjoyed, and I'll talk to you soon. Bye-bye.